So that 90-day rule is for the application of raw manures in the field, where if the product is on stakes, I have to wait at least 90 days before I can harvest. This is for pathogen die-off. And any products that are grown on plastic or on the ground, melons, classic example, I have to wait at least 120 days from the application of these basalls before I can harvest my my crop. And one of the reasons for this is, but while these are nutritious for the soil, since they are animal waste, they can contain human pathogens. These untreated manures or untreated waste products can in fact harbor pathogens. And in the next talk, I'll give you some prevalence numbers that we did when we did some surveys around the Southeast. One way to get around this time of entry into the field is having some type of treated basal. And again, there are a lot of different commercial forms of this, things like fish emulsions that can be heated up to a certain time and temperature. In this case, maybe 80 degrees C for a specific time period. Bone or feather meal, again, that could be heated or steamed. The one or the most common one is just composting, which is just allowing the natural microbial activity within a pile or windrow for those microbes to increase the heat in the center of that pile or windrow. And again, that prolonged heating activity will inactivate the pathogens that may or may not be present. One of the last ways, again, is taking this weight material and then heat treating it and making a pellet out of it. One of the classic ones, and the one that you'll hear about later from Dr. Kharel, is heat-treated poultry pellets. All of these processes are done to reduce the potential for pathogenic microorganisms. And even these treatments, while they're antagonistic or deadly to the pathogens that we're talking about, don't reduce the amount of microbes in the basal. There are plenty of microorganisms left over, even after much more rigorous heat treatments than these. Again, we've tried to kill all the microorganisms. There are things that are just really tough to kill, but our pathogens are fairly easy to inactivate under these somewhat mild heating conditions compared to what we can actually expose the basal to. One of the other reasons for treating, other than knocking down the potential foodborne pathogens, is a reduction in the seeds associated with weeds, and this keeps the amount of weeds down. As you know, most of you who are farming, weeds are an inevitability, and the more you can reduce, again, the less you need to either do hand weeding or, again, in some cases with conventional, use some type of herbicide to knock down the weed population. The presence of a foodborne pathogen in these type of treated basals is very low. Again, these heat treatments, while again fairly mild by most standards, are going to be adequate enough to inactivate the pathogens of concerns. And just to kind of throw out a couple names, in bovine manures or dairy, those typically we look for things like pathogenic E. coli, and I'll provide numbers later, but it can also have things like salmonella. In our poultry litter, those products, typically we look for the presence of things like salmonella or campylobacter. Those are two poultry-associated pathogens that, again, when we look, we can't find them in sufficient numbers. But again, as long as we're treating them, again, we tend to knock these down fairly easily. Now, untreated manures or improperly composted manures have a potentially higher risk. Again, I'll present numbers in the next talk that shows that when we look at cow manure or poultry litter or pig slurries, and we look for these particular pathogens, we can find them. It's almost, in the case of finding E. coli's in bovine manures, I think every farm we tested, and we were testing in the middle of piles, on the outside of piles, and while not every pile was positive for a pathogenic E. coli, at one point where we tested multiple piles within a farm over multiple times during the year, every farm came up positive at least once. So these pathogens are fairly common. And again, having untreated manures does raise that risk, especially if we're not waiting that defined period of time. Only problem is we don't know what that defined time period is. In the case of the produce safety rule, excuse me, there is no defined time period. As a matter of fact, it's left blank or reserved until further research has been accumulated to where they can make a recommendation. And again, remember, this was signed in law in 2011. We've been inspecting on this, or at least the inspectors have, since around 2016. This is being enforced, and now being 2025, we still don't have any guidance other than the recommendation is to follow the USDA NOP standards. And again, that's that 90 and 120 day standard that is currently out there. So again, we talked about this just briefly. These sows contain or can contain things like salmonella, pathogenic E. coli, and one of the most common ones we look for is E. coli 0157H7, and that's just a particular strain. It actually borrowed some of the genes from Shigella, which is where the Shiga toxin comes from. So Shigella lent E. coli some of its toxin-producing genes, and now this particular strain of E. coli, as well as several others, has the ability to produce these Shiga toxins and makes them highly pathogenic and very dangerous, especially to at-risk groups like young children, the elderly, pregnant women, and people who are immunocompromised. So these are fairly common. Like I said, in the next talk, I'll share some numbers of how often we found these and the litters we looked at. But the thing we're worried about is if we were to apply these products to fields, what is the likelihood of these organisms making it to our edible portion of the crop we're trying to grow? And that's kind of the nature of all this. How long do these pathogens survive in the environment? And then finally, what effects do the soil amendments have on the survival characteristics? And some of them are not as obvious as you would think, but again, we'll go over that in the next two talks. So why use untreated basals? Here's one example of a commercial composted broiler, and this is an actually treated one. You have some type of, poultry operation, some hen house. It goes through maybe a front-end loader, goes into a truck, gets offloaded with another loader into piles or windrows. Those can then be loaded up again, other trucks. And there's just a lot of activity, a lot of hands, and a lot of people who have to get paid. So a treated manure product, depending on what operation is doing it and who's doing it, especially if you're not the farm creating it, again, could raise the cost of these products. And while composting is safer, can be a lot more expensive. If you're doing your own, obviously, as the illustration shows, there's just a lot less hands touching it. If you're, a mixed farming operation and you have poultry and you have a veggie crop and you're making your own, say, poultry litter, this saves you money, but again, it still takes man hours and equipment and dedicated sites with, a certain amount of acreage dedicated to this composting activity. Again, costs less money than if you're purchasing from an outside vendor, but it's still, it just still costs a little bit more money. But again, a little bit safer than the uncomposted product. So why use untreated? Again, especially if you're getting it from farm or from a neighboring operation. you have less time, less labor, less fuel. This just costs a lot less money, and that's a plus. Uh, and the National Organic Program gives you that 90 and 120-day guidance. So, yeah, so it's raw. All I have to do, during my fallow season where I'm not planting, is just make sure I meet those guidelines. I keep my proper records, and I don't have to worry about it. And I save a ton of money. And the National Organic Program, the NOP, says this is fine. And this is the crux of all this. So the question is, are those 2 waiting periods adequate to inactivate all the microbes that we're concerned about that we're finding in these products? So one of the reasons why we use untreated, just cheaper. Provides the same sort of nutrient material and may actually provide a little bit more nutrients. And I'll get into that in this slide right here. So when we're using any type of untreated Basal, the fecal matter is deposited. It interacts with the soil microbiota. It is broken down by decomposers and other organisms like nitrogen fixing and nitrifying bacteria in the soil. These are broken down to produce nitrites and nitrates, which are then used by plants. Occasionally, we'll lose a little bit of this nitrogen to the atmosphere, but for the most part, we're breaking down some of these compounds and using them directly to support the growth of our crop, which is good. It's also building up a healthy microbiome in the soil. So again, we're maintaining, or at least keeping that topsoil healthy and not depleting it, which is a problem. When we compost, we can actually lose a little bit of that nitrogen that we're so desperately trying to retain. One of the interesting things is one of the offshoots, other than being nitrogen, is things like ammonia are being released because again, they're not being broken down by subsequent bacteria in the pile or windrow. The interesting thing, though, is the production of this ammonia compound, that NH4 and the NH4 plus and NH3, is one of the key factors we'll talk about in the next talk that actually aids in the reduction of certain microorganisms. So in the composting pile or windrow, the production of these chemicals, especially nitrogen, ammonia, helps reduce the levels of things like salmonella. And some of the research that we have shown is when we knock out those denitrifying bacteria and we don't convert our nitrogen to ammonia, the salmonella lives. And as soon as we add those bacteria back into the soil, the reductions that we normally see come back. So it's kind of an interesting process. So while we're losing some of the nitrogen, we're using some of the components to actually inactivate those microorganisms. So it's a little good, a little bad. But again, what we're doing when we're making this compost is making sure we're removing any of those potentially harmful microbes as we're creating this product. If we look at the what benefits this untreated basals have, and this was a study, Bagball et al. It shows that again we're having nitrogen losses from 19 to 40+%, calcium losses and potassium losses again anywhere from 40 to 60%. So fairly substantial loss in some nutrients that we're applying. But again, at this point, we now have to kind of determine whether we're more concerned about the safety aspects or we're more worried about the soil health components. And again, that's going to be the crux of the research we'll be presenting. Other types of things to be considered is the waste management. We're generating, and this is almost 10 years old now, so it's probably more than this at this point, but over 10 million tons of litter every single year. So that's a lot of product. If this stuff is improperly stored and we have runoff into waterways or leaching into the groundwater, what we end up is an overabundance of nitrogen, and this leads to eutrophication of these waterways. And that just means an overgrowth of either algae or other microorganisms, which can end up using up all the oxygen, and then again, any fauna, fish, or wildlife in that pond will end up dying or suffocating because, again, all these algal blooms are, again, using up the oxygen and overgrowing these waterways. We have a similar situation here in Florida where we have a lot of runoff from synthetic fertilizers getting into the Gulf, and that's causing a more than frequent or increased frequency of red tides. But any waterways where you're, again, pulling surface water for irrigation, again, this eutrophication can, again, lead to problems. So just the waste management component of this is also very important. But it's a resource that is can be good for the environment if used properly, but does potentially increase the risk if not composted or treated properly because of the presence of these microbes. So could we kind of split the use of these different types of amendments? Now, if we use untreated amendments, we have lower risk crops, agronomic crops, or crops that don't touch the ground. If we're planting cotton, flax, or something, we're not going to be eating that product. Thus, the chances of that causing an illness are extremely low. So using untreated basaos, again, makes perfect sense for agronomic crops. For things that are grown on stakes or above the ground, maybe like stake tomatoes, again, the risk is significantly lower than for crops that touch the ground, and it may make economic sense to use untreated litter, especially if we have a growing season that's going to extend past that 90 to 120 days, which is what is currently recommended to ensure that any pathogens that were there will die off over time. And again, we'll present data in a little bit to show whether or not that's fairly adequate. The other thing, too, is keeping proper records. If you're going to be doing composting, and I'm assuming those who do that for a living, I saw one person who has been doing it for over 22 years now. Proper record keeping is important to make sure that you're meeting all the requirements for proper composting. What material, all the identifying information, number of turns, the heat, the temperature readings, application date, thing storage, all that information is really important when we're trying to determine the safety of these soil amendments. So what does the actual rule say? It does say that biological soil amendments of animal origin must be handled and stored to prevent contamination of and cross-contamination to produce, water, food contact surfaces, etc. And that just makes sense. You can't have a manure pile right next to your irrigation canal, which is funny because we were down in South Florida and we saw the manure, the windrow piled. It was literally up against an irrigation or flood control canal, which if you did one 90-degree turn and another 90-degree turn, led you directly to the farm. So that water was being directly applied to the fields with overhead spray, and there was no dam or berm between the manure and the water that was going to be used to irrigate the fields. So again, in that particular instance, that's not being stored properly to prevent cross-contamination. It must not contact the produce during application. Again, we want to minimize contact after this, except we're side dressing. And that way, if there is any pathogen present, they don't have the opportunity to proliferate. Now, again, the USDA National Organic Program's 90 and 120-day interval is not written into the rule. This is only recommended, but there's no guidance to say what the actual time period is. So they're defaulting until such time they make their own decision on what that time period should be. For now, they're defaulting to the 90, 120 days, but that could change. Initially, when this came out, they were recommending over 200, 240 based on some research papers. And that was going to drive some of the organic producers a little bit nutty, because if you were to plant a field, then graze on that field, leave it fallow for 4 months, then plant, graze, fallow, plant, graze, fallow, in 4 month intervals, you have a perfect cycle for your produce, regenerating the soil with the animal grazing, and then that adequate waiting time. So if they went with a longer wait period, that would throw that whole timing schedule off, and again, really hamstring organic production. And that was one thing that when they were doing this, when they're coming out with a rule, that was one of the comments. Until there was scientific information to base this on, they didn't want to have these longer waiting periods imposed, because again, that would just basically interfere with the normal farming cycles that happens. Now, I mentioned Contact. This is the grant that myself, Manan Sharma, and Laurel Dunn were part of. Actually, so was Dr. Kharel. And this was a survey that was done, and this looking at farmers and growers' perspectives on their biological soil amendments. This was done between 2023 and 2024, and a paper was presented. Melendez et al. And again, this was presented at a national meeting just last summer. And the results that we saw, farmers concerned with this. And we have about a 52% of the respondents saying that they were concerned with the use of these soil amendments and the potential hazards they can cause. One of the difficulties that we see is that a foodborne illness can have lasting consequences, and the damage it could do to your brand or to your farm can be staggering. If there's an actual outbreak where illnesses and God forbid a death occurs, it can bring an entire company down and actually could lead to legal ramifications if in fact you're found to be negligent. So obviously the farmers were concerned, at least over half, or just over half, were concerned with the potential hazards that these can cause. The interesting thing, though, is how many people were doing what we would consider proper record keeping and monitoring. Now, this number of respondents is less than the other slide. I think we had 30 plus in the other one. This is only 9 respondents. And the vast majority said no. 22% said, yeah, they monitor, but they don't really keep records. 11% were not sure. So, but honestly, they're not sure. I take that as a no as well. And 0 were actually keeping proper records on their soil amendments. Now, this is people who've been in production for substantial amounts of time already. And again, this isn't an older survey. This was just presented last year, and the data was collected, I think, no later than 2 years ago. So you would think at this point in time that this would not be an issue, but it is. And again, this is what's a little concerning, is that the use of these amendments is very, very common. People are worried about what's happening with these amendments, and but they're just not. There's a little bit of a disconnect when it comes to the requirements such as monitoring. And again, hopefully we'll address those in the future or shortly. So to end this talk, both treated and untreated biological soil amendments are permitted and used for fresh produce production, even organic and conventional. Some even conventional farmers will start out with an organic amendment or like a poultry litter or bovine manure, and then follow up with synthetics for all subsequent fertigation. Obviously, the organic productions use maybe untreated in the beginning and then treated for all, side dressing and subsequent fertigation in the middle of the season. There are no codified standards in the produce safety rule at this time. These are still reserved, and they have been reserved since the rule was released, but they are defaulting at very least to the NOP, which is administered by USDA, and that's that 90 to 120 day standard. These basals are a valuable input for soil fertility and soil health. Anybody who studies soil composition knows that we need to take care of our topsoil, as again, it's a very limited resource, and we obviously don't want to run out. If we are going to use untreated basals, we need to make sure that we're reducing the likelihood of contamination, which again is avoiding contact with the edible or harvestable portion of our produce, trying to prevent splash, and again, waiting an adequate amount of time for those microbes to die off. And that's what we're going to talk about in the next talk, is how long do these pathogens actually do survive once they're in the environment. All right, so I'm going to stop sharing now. Any questions on this first talk?
Feel free to come off mute and to ask Dr. Schneider your question
I
directly.
see some of the questions. NOP certification would require record keeping. Yes, for those people who are doing organic farming and following the NOP, those folks are keeping proper records. For those folks who are again not seeking the organic certification or just using them, like a lot of our conventional farmers here in Florida, using the that initial treatment, followed by just synthetic fertilizers afterwards. Again, the record keeping is a little bit on the suspect side. Now, that's not everybody. I mean, the folks who do organic production are really in tune on this, and they've been following the rules. So I would imagine if we had a room full of just those producers, we'd probably have near 100%. But that wasn't the audience when we surveyed. These were just regular farmers who are not under any special organic production, but they were using these organic basals, at least in part, during their growing season.
Hi, Tom. It's Johnny Massa. I was the guy that was composting for 22 years. I still am composting. I was the only composter in the Salinas Valley before the 2006 outbreak that was tested for 015787. So we're farmers that got into the composting business. So we look at it a lot differently. One of the things that we found out is working the material a lot longer, like a 13 to 15 week process where you're constantly watering and turning the product. We're not getting any more pathogen re-eruption. We have no pesticide residues to speak of. And you can check with one of your colleagues there, Dr. Shuping Zhang. Very hard for anything to be growing in our compost. We follow back to the original research back in the late 60s, early 70s, where we have such a rich bioflora in the compost itself that the regrowth of human pathogens don't happen. Or if there is anything that's not detected but viable organism in there, if it does try to grow, it can.
Yeah, John, and to that point, I'm going to show some slides that concur with exactly what you're saying. Especially with salmonella, one of the studies that we did will show that the microflora associated with this sort of rich microbiome containing compost really will outcompete organisms like E. coli. Oh, yeah. When we tried that same experiment with salmonella didn't care. What salmonella cared about was the production of ammonia. And we showed by knocking out those bacteria responsible for the conversion into ammonia that we weren't able to get the reduction in salmonella. So the denitrification that's occurring and the production of ammonia, which again, we're losing nitrogen to the environment, but we're also killing the pathogens, works great. The diverse microflora will be antagonistic to certain things like E. coli, which is also good. But we're also seeing some other things, and I don't want to have any spoilers because Dr. Kharel is going to talk about this, is that the presence of additional nutrients sometimes is counteracts some of the beneficial things that we see with the compost. So while certain things are knocking down the populations, we have certain things that are sort of aiding these populations. So there's a little bit of push and pull going on. But again, as far as composting goes, the stuff with the NOP is really interesting. And the numbers that we're going to show in the next talk, and I'll get into it just in a brief second, the numbers that we're finding, and we find them a lot, and the survival of certain of these microorganisms, we're not taking into the case or taking into account whether or not the numbers that we're finding are clinically significant, which means how much can we get away with without causing illness in the populace? And that goes to something we would call effective dose. We really don't do that anymore. That's kind of we don't measure effective dose because it's different for every person. We would normally do a risk assessment, and then we can calculate the number of individuals who might become ill if they're exposed to a certain level. And a lot of the cases we're going to show that we did see these things survive for extended period of times, but we don't know whether or not those are or pose a risk of illness to the consuming public. And that's going to be a hard question to answer as we go through the next two talks.
Actually, I could probably help you answer some of those questions. With the process that we do a 5-week pathogen reduction phase. So we're doing 5 weeks over 140 degrees Fahrenheit.
And and again, one of the things to also consider, though, looking at the spectrum of people who do composting, we have folks doing static pile, not turning.
Right.
And then we're not getting a whole
issues.
lot.
There's issues with static piles. I agree with you on that one.
And then we have folks like you, and especially here in Florida, we have an operation literally just about 45 minutes up the road from where I'm at one of their research operations, where their windrows are excellent. They're turning them, every week. They have specialized equipment, and they're making a really well-aged compost that is pathogen-free. So going through that entire spectrum of the backyard farmers with their static piles, folks who are doing professionally turned windrows for extended period of time, there is
a huge difference.
Yes,
huge difference. Yeah, no. I mean, I always have to sit there and talk to the schools about them going to. Well, we do our own compost. I said, do you have somebody there constantly watering and turning it during the summer months when you guys are off? And everybody goes, no. I said, well, tell you what, I'll just bring you all you want.
And we'll say this for the end, but the whole farm to school thing drives me crazy because, again, trusting
I agree with you.
a third to sixth grader to do that properly versus someone like yourself or some of the folks here that I work with. Again, there are different levels to this, and I appreciate those different levels. What else we have? Let's see, different environments, more hospitable, less human. The difference in hospitality. What environments are more hospitable? We'll talk about that in the next talk. So, Tom, if I don't answer your questions in the next talk, you can kind of jump in, and I'll try to answer it then. But we're going to go over the different conditions that the human pathogens seem to prefer. So I'm going to if you wouldn't mind, I'm going to table that one until I'm done with my next talk. And then Don, what do you got for me? What do we know about the ability of on-farm to effectively create under control conditions? You just talk about the past. Again, as Don Sockel in the chat just said, the levels of this, there are folks who are more adept at this and have the equipment and manpower versus folks who, are composting, quote unquote, by just leaving in a static pile on a tarp in their backyard next to the barn. And again, not keeping those proper records. Trust me, when we saw the results that only half the folks that we were talking, or less than half, were actually keeping proper records. Again, it's a little frustrating, a little scary, again, because we want everybody to really keep really good records on all this, so they're producing a safe product. All right, anything else for now? So that was the intro. I'm going to jump into some of the research that we've done, and this is all on prevalence and survival of these pathogens. So hopefully I'll answer some of your other questions. And I apologize for having to talk at you twice. I don't even subject my own students to that typically, but bear with me. There we go. I think we're sharing, and then and just give me a thumbs up if we're good. Outstanding. All right. So this is some of the research that's come out of my lab specifically. And again, this is coming off the Contact grant, which is the PI for that grant is my partner in crime here at the University of Florida. You might know her name, Dr. Michelle Daniluk, though. Shout out to Michelle for sending us the resources to conduct this research. Again, we're looking specifically at the factors that promote the survival of these microorganisms, and then what happens when we add or subtract these amendments to the soil. So one, we're going to look at the prevalence of E. coli in bovine manure, the prevalence of salmonella in poultry litter. And again, we can find E. coli in poultry and salmonella in bovine, but the most common things are, E. coli in cattle and salmonella in poultry litter. We're going to look at the survival in the lab. Again, it's a little bit difficult to take this out into the environment. We can't go spraying salmonella willy-nilly in the environment, even on our research farms. Call me crazy, but yeah, spreading pathogens around the environment with potential subject to runoff and wind blowing and dust particles and the occasional hurricane that we have here in this part of the world, not a good thing. So most of the things with the really nasty pathogens we do in-house here in the laboratory, so to keep everybody safe. But what we have done is looked at generic E. coli, and these are the E. coli that are non-pathogenic. About 70% of us who are listening today will have sort of a commensal non-pathogenic E. coli in our gut. Doesn't hurt us, just part of the natural part of our personal microflora. And it behaves very similar to some of the pathogenic ones. Sometimes it's actually maybe just a little bit weaker in this, where if this survives for X number of days, we might even say that the other ones or the pathogenic ones might be there around a little bit longer. And we'll see how that plays in when we get to the end of this talk. And then I'm going to say the good stuff for Dr. Kharel. She was involved in this project with another postdoc, Cameron Bardsley, and that's a matter of fact, that's Cameron walking in that picture there. And then my student who's now at the University of Georgia working with Dr. Dunn, the aforementioned Dr. Dunn, Charles Bency Apollon. Charles was a student of mine, and he's now currently working on his PhD at the University of Georgia. And there's my boy Bency. So what is a BSAO, or a biological supplement of animal origin? Again, this is the amendments coming from animal sources. Again, typically the most common are bovine and poultry. And the FDA, again, as mentioned, is deferring action until a risk assessment has been performed to determine the application interval for untreated BSAOs. Now, if you have a composted litter, like we were just chatting about, one that properly turned, heated, all the things that you need to make proper compost, there is 0 day of entry. You can you can side dress that stuff down and go in the field the next day and harvest away. There's no time period associated with that under the current regulation. For untreated BSAOs that have not been composted or treated in some fashion, there is no recommendation at this point other than the 90 to 120 day NOP standard. Now, this does not guarantee anything that even if you're using the NOP, that you'll be safe from litigation. This is until we have a better plan, this is the one that FDA is kind of relying to. As soon as they get any potential information or any indication that this is not functioning properly, they reserve the right to change the produce safety rule. Now, they haven't done this, and it's been better part of a decade now. So, but we are still waiting. And part of this research was funded to answer those specific questions and to try to help them make a decision of whether or not what we're seeing in the environment is going to affect public health. And the studies that we're talking about were done in conjunction with folks at University of Delaware, University of Georgia, UC Davis, University of Arizona. So a lot of universities were involved in this project. And this particular one, where we're looking at survival in the fields, we did side-by-side comparisons with the folks up at UGA and their farms up in Georgia. But let's just go through some of these. First off, the number of foodborne illnesses associated with fresh produce is increasing or is pretty much stable. And this goes back to like the 1970s when we decided, hey, fast food just ain't cutting it. We need to eat more fresh fruits and veggies. And when we started to have healthier diets, more salads, we saw a corresponding increase in the number of foodborne illnesses, and the number of produce associated foodborne outbreaks also seems to be steady or increasing. And these numbers kind of bear out. If we look at what has caused these illnesses, we look at organisms like salmonella, which was very common in crops like tomatoes here in Florida. We were having outbreaks in the the late 1990s, early 2000s, and we were having them one or two a year before we put our tomato good agricultural practices program in place. E. coli we see a lot in leafy green products, and this goes back to the 2006 spinach outbreak in California. This culminates in the release or the creation of the leafy greens marketing agreement, which again played a huge factor in the produce safety rule development as well. Initially having the same water use requirements, which have been eliminated now. But these are two common ones. So these are the ones that we concentrated on. There are about 30 pathogens that we see over and over again in food. It doesn't mean that we don't have an esoteric one or a rare one every now and again. But for the most part, we see a large number of salmonella outbreaks and E. coli outbreaks because these are fecally associated pathogens, which means if an animal were to contaminate or have any droppings in the field where we're producing our product or growing our product, it can become contaminated. It's also one that, again, can be passed from humans who are not washing their hands onto the product itself. So while there are a number of pathogens for this research, we're only going to concentrate on these two organisms, which are most common. Now, just for some numbers, salmonella will cause about 1 million to 1.5 million cases of foodborne illness every year, mostly associated with poultry products. E. coli, maybe 80 to 100,000 cases per year. And again, we see this mostly with undercooked beef products. But then again, when we have the use of any of these soil amendments or, irrigation water, where the runoff will contaminate irrigation water for an adjacent farm, we have seen produce become contaminated. And some of the classic cases most recently, that the big one was the 2006 spinach outbreak. But we can go back to the romaine outbreaks of 2018, 19, and 20, 3 years in a row in Yuma in the Salinas Valley, where we saw pathogenic E. coli being the cause of those outbreaks in the leafy green. So these are some of the things that we're looking for. So the first thing we wanted to do is to figure out what the prevalence was. So we did a survey in Florida, and actually some places in Texas, believe it or not. But for the most part, these samples were taken from Florida, and we would look at these piles or windrows, and we weren't looking for the presence of just E. coli. We were looking for the presence of these Shiga toxin or these S-tech E. coli. These are the pathogenic ones. And then one particular strain, E. coli 0157H7, whether or not it had Shiga toxin genes in it or not. So when we were looking on these farms, if we were to look for E. coli, we'd find it 100% of the time. But we were looking for the pathogenic types, and looking at whether we're seeing it on the surface, in the depth, where we're seeing it in ponds, lagoons. So in this particular study, we were surveying raw manure and just looking for the presence of not generic E. coli, but E. coli that can make you very, very ill. And here's some of the numbers. If we look at the presence of any STX at all, around 6% in the piles. In lagoons, where we have a lot more water, we're not having a whole lot of desiccation. It was up to 11%. In the barn, again, a little fresher in this case, about 13%. And then when we had a screened solids, maybe around a dairy operation, slightly less, around 8%. And then we broke it out into whether it had this SCX gene number 1 or gene number 2. And in this case, a little lower, because some of these would be in one, some would be in both, some would be in neither. And then we look just for the pathogenic. And again, we're looking at upwards of, in the piles and the lagoons, over 20%. So 1 out of 5 samples. And we went back to the farms multiple times. So not all piles would test positive, but we found at least one positive pile on every farm that we looked at. So if you're looking, we took in the piles, we took almost 200 samples. And again, we're getting a prevalence rate of about 20%. And at least every farm had at least one sample that tested positive. And again, this is not saying that we have these in sufficient number to cause illness. We just now know that they're present within this within these operations. And again, just want to be concerned, because if they get into a product or a cull or some damaged fruit, there is an opportunity for these things to increase. Now, over time, as you compost or as these things age, these numbers for these particular organisms, which like being inside an organism, like a body or a cow or a person, will tend to go down once they've exposed to the environment. Typically, they don't multiply in the environment unless there's an abundance of food or they re-enter another host, another animal. The food sources, typically damaged produce or cull produce, is one of the easiest area where these things can proliferate. So again, this is sort of the risk associated with bovine manures. We'll take a look real quick now at what we saw for the poultry litter, again in Florida, when we went sampling, and these were around poultry operations. And we also looked in this particular study, not only presence or absence, but how much was actually there, and whether or not these were antimicrobially resistant strains of Salmonella, which again, antimicrobially resistant strains can pose a problem, because if you become sick and you need to be treated with antibiotics to become healthy, if these organisms are resistant, your first line of defense may be ineffective, because these have become resistant over time. So if we look at some of the numbers here, and if we look at the prevalence numbers, this is, I would say shocking, but we're getting near 50% or greater, depending on the months that we're testing. Now, interestingly, what we're seeing in the environment, this is out in the wild now. These are actual piles on the ground or in the farming houses. We see much higher prevalence in the winter, and this will repeat itself as we look at the data. These organisms tend to survive better in cooler environments, going back to the question that we had in the chat. And as it gets hotter, whether or not we're having increased desiccation, potentially we're having the conversion of the microorganisms responsible for breaking down the nitrogen into compounds like ammonia, which are bacteriostatic or bactericidal, increase, or we have the increase of other bacterial predators within the soil, that microbiome that's helping us compete. And all those organisms just happen to be more active in the summer. So in the hotter months, we see the shorter survival or less prevalence. In the cooler months, we see the largest prevalence, and then the spring season is somewhere in the middle of all that. But on average, you can find Salmonella about 61% of the time, which is crazy. Now, if we look at the amount that we're finding, and this was done by MPN, our most probable number. So this is rather than plating the litter or the slurry that we make on a plate, we have to do a multiple tube reaction in order to pull out the number. So again, there's a little bit of microbial gymnastics being performed here, but it allows us to detect very, very low levels, and it also allows us to detect injured cells. And if you look at this, the numbers are very low. Less than an organism per gram, which means we're getting like four organisms per 10 grams of soil or slurry that we're testing. So the numbers that we're seeing are very low. And this one, it goes back to my comment of whether or not these are clinically significant numbers. Now, if I have something around 0.2, I'm getting a positive right here, 0.2. Hopefully you can see my mouse. The clinical impact or the ability or the likelihood of that causing illness is going to be extremely low. But once we get to levels that are a little bit higher, then the chance for illness increases as the risk to the consumer goes up as the dose or the amount that they're consuming is going to go up. So we're finding, whether this be pathogenic E. coli or in case of salmonella, we're finding these things fairly often in the environment. And again, without proper composting, these things, again, especially the raw manures, have a greater opportunity to make it to the consumer. So let's look at some of the studies. For the in-lab studies, we were looking at the effects of moisture content. So does heavy rain or excessive irrigation, is that going to change the way these organisms behave? Common thought was, well, if I have flooding events or really moist environments, they're going to be happier versus if I'm irrigating or the rainfall's giving water levels that are close to wilting. And these percentages, 15, 25, and 35, are for a loamy soil. We also did another study, which I'm not going to talk about today, which I believe was 5, 15, and 25, which is for a sandy soil. Didn't have the same moisture holding capacity. And then we also did 2 different storage temperatures. One, which is a more temperate spring or fall, 20 degrees Celsius, versus a hot summer day, which is about 30 degrees Celsius. So looking at the different environmental conditions. 2 different types of sand. Again, the sandy soil and the sandy loam were 2 different areas. These were about an hour east and an hour north of the University of Florida. The analysis for these, you can see slightly more clay in the loam. Again, also that helps with the water retention. The organic material was much higher in the loamy soil versus the sand, which makes sense. In Florida, we have a lot of sandy soil, which again, not a whole lot of organic matter associated with that. So adding something like a biological soil amendment does help keep that topsoil a little bit healthier. In regards, even our sandy loam is pretty low in organic. What we would do is we'd actually get sand from those 2 centers. We'd make 1 kilogram microcosms, and then we'd grow up a 5 cocktail strain of salmonella. So we took 5 different strains that were associated with produce outbreaks. We'd grow those up. We'd spit them all down, and we make a nice inoculum, and then we inoculate the soils. And then we did the same thing with E. coli. We only used 3 strains of pathogenic E. coli, which is an 0157, which is the particular nasty one. Another one, 0104, and then 0145. And these were also produce-related outbreaks. So 3 pathogenic strains of E. coli, 3 pathogenic strains of salmonella. They were added separately to the little soil microcosms, which were kept in bags and then kept in incubators. The moisture remained constant because the bags were sealed, and then the oxygen would be replenished every time we'd pull a sample. And this is what we found. We were able to find at 20 degrees that these organisms, in this case, E. coli on the left, salmonella on the right, would survive just fine, whether I was in when in the loam at 20 degrees. We were able to detect them out to 200 days, which is exceeding that 120 days set up by the NOP. And the question that we have to ask, though, is that 120 days, or about 1 log CFU, or that's 10 colony forming units per gram of soil. That's not a whole lot of microorganisms. Now, I don't know if we want any of these pathogens at any level to get to our food product or potentially transferring to our crop. But what it does show you is that after that initial die-off phase at 90 and 120 days, the vast majority of these organisms will die off, even at these relatively cool temperatures. And again, whether or not these numbers that we're seeing post 120 days, whether or not they're clinically significant or not, is a whole nother question. And I'll leave that with people with higher pay grades than myself to do the biostatistics on that. But again, we were able to recover either E. coli or Salmonella all the way out to 200 days. And in fact, if you look at the moisture content, they really don't care. Matter of fact, in many cases, the only difference that we saw is they survive better in the drier than they did in the more moisture soil, which seems contradictory. You'd think they would desiccate and die off quicker. They did not. If anything, they were the same. And when we did see a difference, the drier ones actually survived better. And again, I'm scratching my head on that one, but this is what we saw. Now, if we did the same experiment and we raised the temperature to a summer-like environment, this is what we saw. And you can see a dramatic difference here. So if you look at these, we see rapid die-off. Between 50 and 80 days, we're getting to near unrecoverable levels. The left one is in a sandy soil, which is a little bit drier, doesn't hold as much water, but we can still look at the 15 and 25 as overlap compared to the 30 degrees of E. coli. And at 30 degrees, we have a very fast die-off compared to the 20 degrees. Whether it be loam or sand, we saw this rapid die-off, but in the loamy soil, we're able to recover it past 100 days. Again, extremely low numbers, but we're still able to tease it out of the background soil, and even up to around 250 days. So the one thing that we see is pretty universal through all the studies is that when the environment is cooler, we tend to see longer survival. We tend to see things die off faster in warmer conditions. But we also saw that the presence of higher organic led to longer survival. And that's going to be more evident when we look at some of the work that Dr. Kharel is going to talk about with Bensie's work. Now, we looked at naturally occurring E. coli. This E. coli was generic E. coli, the non-pathogenic kind that was recovered from our bovine manure. And then we put this into microcosms. So we took naturally occurring just E. coli that we're seeing out in the fields, and we put it into a microcosm. The sands, the live oak sand and the citra sand. Live oak was the sandy soil, and the citra was more the loamy sand. And then we tracked survival. Now, this work, by the way, there were two young ladies. One was Christina Underthin, and the other was Samantha Chekic. I don't want to credit them for the work that they did. And this one's kind of fun because if you look at this, especially in those loamy soils at the cooler temperatures, we had survival up to 420 days in the lab. So this is greatly past the 120 days. We're literally at our limit of detection for this type of test, which means we're really just testing presence or absence. We're not able to tease out a whole lot or plate a whole lot of numbers out, but we're able to find these organisms well over a year after we inoculated them. We took up 2 incubators. We monopolized them for well over a year. Matter of fact, the young lady who started this work graduated before they died. So basically, the E. coli out-survived the student. The student already left and had a job by the time these things finally faded out. And then I have to thank one of the other students I'm going to be talking about, Alan Gutierrez. Alan actually picked up the mantle, the baton on this one, and carried it through to the end. But if you look at it, the 20 degrees, that's these orange and green lines. Hopefully you can see my mouse. here. They showed a little bit better survival after 90 days, whereas those higher temperatures, the blue and the red lines, sort of bottom out at about 60 days. And then we're just able to get maybe a positive or negative. We're at that limit of detection. Whereas the cooler temperatures, we have better survival. But again, at these temperatures, we were able to detect viable microorganisms out to 420 days. And just for what we're not showing on this grant, is the time at which these things are sort of reaching that limit of detection for these tests. And again, one after about 80 days and one after about 180 days before they go to that point where we're just getting a presence absent. So they are amazing at surviving at long periods of time. And what's not shown is the controls here. All our controls were negative. So we were running controls side by side just to make sure that we weren't getting any naturally occurring microorganisms, and we weren't. When we put the soil amendments or the E. coli from the soil amendments in these microcosms, they were able to survive for extended periods of time. All right, let's go to the field now. And again, this is specifically Samantha Chekich's work. And in this case, what we see is we have plots that are 10 feet by 30 feet. We spread about 6 tons of manure. The first study we did 2 fields for 2 seasons over a 2-year period. Each field would take about 6.5 tons of manure to reach the nitrogen rate that we were trying to meet for this study. While we're using a front end loader driving backwards so not to contaminate each of the plots. The first ones we actually did by hand. So spreading 6 tons of manure by hand. Yeah, if anybody's sprinkling up their nose right now, please feel free to do so. It was that bad. But yeah, we got a little smarter in year 2. These were the plots. You can just kind of see. We'd spread them out. And again, we had 3 control plots with nothing in them, 3 with very light application of manure, 3 with the agronomic rate, and then 3 at double the agronomic rate. Again, 2 locations, 2 seasons, and then there were 3 replicates for each one of the trials. And this is what we saw. Again, they were 3 by 10 meters. These were taken. These were screen solids taken from a dairy operation just up the road up in up Live Oak, Florida, and this is our research center up at the North Florida Research and Education Center in Live Oak. And we tracked the naturally occurring E. coli, and we monitored for 300 days out in the field. Again, close to a year. And this is what we saw. For the light, medium, and heavy, statistically, we weren't seeing a whole lot of difference between them. Again, there are a couple of spikes here and there, but for the most part, we were able to recover these things. And again, we're at the limit of our detection, which is about 10 to the 2 or 100 microorganisms at this point. You see that we saw occasionally as we would sample these things, we'd hit a hot spot. And we try to correlate this to potential weather events where we looked at solar radiation, wind, rain, temperature, and just total rainfall. And we got 0 correlation with any of the environmental factors. And the only thing that we saw was the seasonality had a difference. Now, the other thing, because we looked at the potential for solar radiation knocking down the microbes at the surface, we also took core samples. I forgot to mention, at each one of these plots, not only did we take surface composite samples, we also took composite core samples. And again, this is the work of Samantha Chekich, and again, bless her heart, this was a lot of work. These were hundreds and hundreds of samples. And we were able to detect these, at, albeit low numbers, well beyond 120 days. So again, even letting this sit for the NOP standards, we have these numbers that, while we don't know if these would be considered clinically significant, we're able to find these microorganisms in the environment. And again, you just kind of see the vast majority is dying off within the first 30 days or so. And you'll see this with the next study as well. But yeah, the vast majority die off. And then we have the things called persisters. And that's what they do. They persist for very long periods of time. And this is what sort of concerns not only us doing the research, but obviously FDA is concerned about persisters as well. And there's a lot of other schools doing similar research. Field conditions, same thing. Now we're looking at summer. And you notice, again, warmer weather, much faster die-off. So we're in the previous slide, I'll jump back real quick. We're finding these things out to about 280 days. And when we found a negative, and this one, we didn't hit any negatives. We found positives all the way through. This one, we had them dying off around 130, 140 days. And complete die-off or extinction was determined by having 2 sampling periods that were negative simultaneously. So we might have had a negative one here, but it bounced back. And then here, and then we had two negative ones afterwards. So again, the vast majority of this die-off is occurring in the first 30 days. They would persist, but not as long as we had in the cooler weather. So we're going back to what we saw in the microcosms. The one factor that seems to correlate very well is temperature, and whether or not this is the organisms slowing their metabolism down, or we're affecting the breakdown and the production of things like ammonia that would help drive these things down, or we're slowing down the other predators because of the lower temperatures. More things are active in the summer, and we do get better die-off in these warmer weather, and that's kind of what we're seeing in both of these studies. The effect on the microbiome. So this is, we're looking at, again, does microbiological diversity have anything to do with survival? And then what we did was we took our soil and we autoclaved it. And for those who don't know autoclaving, it's 251 degrees at around 15 PSI. So we're basically trying to kill everything off. That's how we sterilize our glass when we're doing research. These would be autoclaved for 90 minutes, which is greater than the 15 minutes that we would use for a microbiological experiment. And we did it 3 times sequentially. And even then, the population of microorganisms that can survive those heat treatments was dramatic. And we still had plenty of microorganisms. Not the same. We killed most of the easily killed microorganisms, but there was still some microbiome left, just a different species. But when we removed some of these easily inactivated microorganisms, what we call vegetative or actively growing cells, the autoclave cells, when we reduce the biodiversity, they survive better than the natural soil. So in this case, those organisms within the soil were having an effect that was detrimental to survival. So the more diverse the soil is, the faster die-off we had. When we autoclaved the soil, excuse me, we saw better survival. So again, by reducing that microbial diversity, we actually aided in the survival of the pathogen, which is, again, ours the case that a biological soil amendment versus maybe a synthetic would be helpful in knocking down the numbers for the most part. But even so, with these autoclave samples, and these were with pathogens done in the lab. These are the pathogenic E. coli. Again, they would survive to our detectable numbers, presence, absence. We below 10 to the 1 after about maybe 70 days or so, but we're able to detect presence, absence. Sorry that keep going. My mouse keeps jumping around. Out to about 165 days for some of the strains that we looked at. So microbiome does have a huge effect on what we see. And you can basically see, again, autoclave versus non-autoclaved. And if we look at the sheer numbers, at 28 days, I'm getting almost complete removal. Actually, I'm getting very little removal in the first autoclave in 30. but under the natural conditions, I'm having almost a 5 to 6 log reduction. So there's a huge difference after 28 days. When we cool the soil down, we don't see as big a difference. And again, it goes back to what we've been seeing in all the previous experiments. In warmer weather with a heavier microbiome, we're seeing a lot, pretty much the same reduction, 6 or 7 log reduction. But when we cool the soil down, we see better survival of these pathogens. So going back to the original question in the chat, what environmental parameters seem to affect things the most? What we see the most is temperature of the soil. And we do see that these survive better in cooler temperatures. Now, when we did salmonella, and again, we learned our lesson in this one, instead of autoclaving the soil, we irradiated, we got rid of everything. We made it basically sterile. We irradiated with about 25 plus kilograms, which again, pretty much enough to kill anything. And we would figure that the salmonella would last longer in the natural soil or die off quicker in the natural soil. Annoyingly, salmonella did not play along and didn't care whether it was irradiated or not. So unlike E. coli, salmonella behaved quite differently. And matter of fact, didn't care whether there were other microorganisms in the soil. What did happen, though, with the salmonella was the production of ammonia. So as we composted the soil and we broke things down, we would convert the urea in the poultry litter to ammonia, and we can monitor the production of ammonia in the soils. And again, you see the increase of production of, depending on how much of the substrate, in this case urea, and how much of the enzyme, urease, and how much of the ammonia would be released. And we could check that. And when we stop the production of ammonia, what would happen would be the salmonella would survive. As soon as we return either urease or an organism, Corynebacterium, when we added that back, so it would produce ammonia naturally to the soil after it was irradiated, we saw the die-off again, and we saw the production of ammonia again, and we saw it die off. So salmonella seemed to be affected more heavily on the microorganisms being present to break down the nitrogen in the soil product and resulting in the soil basically acting as a sanitizing agent because of the production of ammonia. And we saw a direct correlation between, again, the production of ammonia in these microcosms versus the survival of salmonella. So what appears to be 2 different mechanisms for the 2 different organisms, but again, in both instances, composting helps break down the microorganisms, though the presence of that organic matter is going to play an important role in the next couple of talks. So I'm going to wrap up here. 1, we know the prevalence. We can find these pathogens in biological soil amendments. They're there. You go looking for them before they're properly composted, you're going to find them at a very high frequency. Not all of the piles you're going to find are going to have pathogens in them. But if you have multiple houses or multiple herds, the chances of you finding them, again, pretty good, because we went to a lot of different farms and we were able to find them in every single farm we went to eventually. Composting, proper composting can eliminate things like salmonella and E. coli. If done properly, these things are not impossible to kill. But again, without proper composting, they can survive for extended periods of time. And again, in the lab, we found them, up to 400 days, but even in the natural environment, 100 to 200 days was not uncommon. The presence of amendments, again, the biological source within the organic material within the amendment, again, seemed to aid in the survival, though the complexity of the microbiota seemed to aid in the die-off. So there's a little push and pull going on here. One thing, the big take-home point here is that when you're looking at lab studies versus field studies, and we've done both, lab studies behave differently, way differently, and the numbers tend to be longer. Again, you don't have as much variation. The temperature doesn't cycle up and down. You don't have other types of impacts like solar radiation. drier desiccation occurring during drier days. So whenever you're looking at a lab study, you will tend to see exaggerated survival numbers. And one of the reasons that we go into the field with non-pathogens is so we can get a better idea what's happening in the actual environment versus the microcosms. We just realize when you're reading any scientific paper that when we're doing stuff in the lab, it's going to look different, and we know that. But even the stuff that we've done in the field with some of our what we would call surrogates or non-pathogenic strains, we do see extended survival. Now, we didn't show any of this data, but we did track soil aeration, wind, rainfall, soil, and air temperature. And the only thing that was correlated with die-off was air temperature, and for that matter, and soil temperature, because they went hand in hand. So again, if it was hotter out, we saw a faster die-off. When it was cooler out, we saw extended survivals. And the big question, as I mentioned a couple of times, is we see the vast majority of these microorganisms dying off in the beginning of, say, the composting, or just sitting, after they've been excreted by their animal host. Within the first 14 to 30 days, the bulk of these microorganisms have gone through a 4 or 5 log reduction. And that means like 99.999% reduction. So the vast majority of these organisms dying off, we're still able to find trace amounts in the environment for these extended periods of time. So with that, sorry to go over a little bit. I'd be glad to take any questions on this particular study. I'm going to stop my share so I can see the chat. What do we got?
One is around copiotrophic versus oligotrophic microorganisms.
Yeah, Tom, that's above my pay grade. I apologize. I'd have to get back to you on that one. The diet controlled STEC question, there's plenty of evidence that that diet can change the prevalence, whether they're on feed or grass. And I know a couple of producers will switch into pasture to change the gut microflora in the cow, and we do see lower levels of pathogenic E. coli depending on diet. And that would be one of my animal science colleagues got to weigh in on that. Soil micro communities operate differently in the presence or absence of plant roots. Yeah, so one of the things, Tom, that Karuna is going to be talking about is we did a study that we did side-by-side inoculation with plain soil and then onions planted as a crop alongside, and we looked at the presence absence. We didn't see a difference, and she can go more into that. One thing we found out, composting the single feedstock. So John is saying that when there's more biodiversity, when they're mixing feedstocks, that you are seeing faster reduction, potentially. We've never done work like that to see whether or not that aids or hinders. I have, like I said, if you're seeing that, that's great. We do know that when we get rid of the diversity, things will survive longer in the lab.
Yes.
Yeah, and then
yeah, definitely, Keith. I mean, we've had we've done our own cause analysis on this, and we've found huge, just even adding 2 or 3 different types of substrates. So all of our compost now are all blended, mixed feedstocks, and then we don't do a single substrate like we used to do before in the past because of that. And the survivability is gone. We pulled 12 samples per windrow. And our batch could be anywhere between 10 to 12 windrows. And Primus Labs does all of our testing, and we literally have if there's one bit of microorganism whatsoever, it's a fail. So it's definitely a huge difference. And then also working the material longer. And I think that's one of the biggest problems that we see in the industry is that people don't work the material long enough. It's that.
Yeah, there's plenty of research. I know colleague Trevor Susslow has done a lot of work on composting out at UC Davis.
Oh, I've worked with Trevor many years.
So it's funny, when I ever lecture with Trevor, I always feel like a Sunday school teacher lecturing alongside the Pope. So so if you've been working with him, you know all about that. Trevor's done some really great work with with composting, and he has a lot of great papers out on how long you work that and how you turn it and all that other good stuff. Everything is everywhere. It's the environment that chooses. Yeah, sort of agree. Again, for me, especially being in Florida where we have hotter weather, we don't see it as often. One thing we worry about is some of the cooler environments, say like in Ohio or in the Delmarva area, which is the Virginia, Maryland, Delaware area here in the US. We're finding that some of the pathogens may survive longer because of the cooler temperatures that they have compared to Florida. And again, we've seen some natural occurring things like salmonella being more prevalent up there than when we find it down here. But yeah, if you look hard enough, you're going to find a pathogen. We've checked the waterways and we pretty much find things everywhere. And then the last question by Kelsey, have you looked into compost teas and if they're beneficial or detrimental to the organisms? We have not looked at teas specifically. You'll see from Karuna's work, we've just taken small amounts of our fully composted poultry litter, heat-treated poultry pellets, and then compared them side by side with just plain old synthetic, and then looked at with and without plants, because they mentioned how they behave in the presence of plant roots. And you'll see, again, with the work that we've done with onions, it didn't make a whole lot of difference in our case. What we do see again is that temperature has the biggest factor. And again, the folks at the University of Georgia are preparing a paper, and Dr. Kharel and our group just finished publishing one where we looked at some of the environmental parameters surrounding Bensie's work. And again, honestly, the one that I see over and over again is temperature. Some people will see rainfall, but that could be just location dependent. Anything else? These are great questions. And I'm going to have to look up oligotrophic versus copiotrophic. I don't know what the question is.
Dr. Schneider?
Yes.
Dr. Schneider? Okay, so one of your colleagues down there at University of Florida, Ariana Van Bruggen, who's retired, she's the one that came up with the importance of copiotrophic versus oligotrophic environments. And copiotrophic just means a very nutrient-rich environments, like high nitrogen and high soluble carbon. And those kind of environments seem to allow human pathogens to hang around longer than in much more nutrient thrifty environments, where it takes microbes harder work to get at that kind of nutrient. And so she did some really important work on that, I would say, probably back in the 90s.
Yeah, so Dr. Van Bruggen, who again used to work here, I think she went to Wagner University, but yeah, she retired a couple of years ago. And what you'll see from Dr. Kharel's talk is when we add, again, these organically rich components to the soil, we can actually increase survival. When we add microbiome, we can actually negatively impact. So there's a push and pull going on here, which you'll see in the next talk. So yes, having nutrient rich can actually help these survive longer, but having a rich microbiome can help die off. But if you take something that's rich in nutrients, like poultry litter, and then you heat treat it and you get rid of the diversity, now you have just plain old biological enhancing, nothing to help kill it off. And then we see, oh, well, again, spoilers. I'm going to let Dr. Kharel tell you. I won't spoil it, but I'll give you a little teaser. We found some interesting results, Tom, and I think it's going to answer or at least feed into that question. And thank you for that information, by the way, Dr. Arena Van Bruggen's work, who, again, enjoying her retirement right now.
All right, thank you for the segue. Our next speaker, like Dr. Schneider mentioned, is Dr. Kharel. She's an assistant professor with Louisiana State University, and her research focuses on microbial food safety and quality, with an emphasis on microbiology of low moisture foods and fresh produce. She's also investigating the persistence, survival, and ecological behavior of foodborne pathogens and exploring effective post-harvest treatments to mitigate the microbial risks in food and food contact surfaces. Additionally, she's also looking at evaluating the impact of such treatments on the quality and shelf life of food.
Thank you, Arpana. Let me try to share my screen here. Is that good? Perfect. All right. So good afternoon, everybody. It's a pleasure to be here today with so many dedicated growers and professionals who play such important role in California's and to a large extent nation's agricultural landscape. Thank you to the UC Extension and Aparna for inviting us to speak. And of course, thank you all for being here. So my name is Karuna Kharel, and I'm currently an assistant professor of food microbiology at LSU Ag Center. And today I'm excited to share with you some of the research that Dr. Schneider has already given a lot of teaser about that we conducted at the University of Florida, where I worked as a postdoctoral associate with Dr. Schneider for 2 wonderful years before joining in this position last fall. So here our study focused on the survival of enteric pathogens in soil amended with treated biological soil amendments of animal origin. And I'm also going to be referring it as basalls. So while treated basalls are considered safe, and as Dr. Schneider mentioned, that they are permitted for use under regulatory guidelines, the reality is that mishandling or if there has been any unintended contamination in the field, which can still occur, how does that come into play, right? Our goal was to understand how these factors influence pathogen survival and its persistence in agricultural soils. So in this presentation, we'll walk through our study on soil amended with poultry-based treatments, specifically the composted poultry litter and heat-treated poultry pellets. So our goal was to determine how E. coli survived in these amended soils over 2 growing seasons. So we conducted this experiment from November through April, which is usually the colder and the coldest months here in the South, in both 2021 through 23. Beyond E. coli survival, we were also interested in understanding the potential transfer of these pathogens to the crop. We know that raw fresh produce is exposed to multiple contamination risk during harvesting, post-harvesting, processing, transportation, and even during final preparation. So we wanted to understand how that pathogen can also transfer to the crop. While there are several produce-associated salmonella outbreaks, very well documented, and a lot of recent data suggest that there is an increasing trend in the outbreak in fresh produce. Interestingly, outbreaks that are linked to bulk vegetables, right, like onions, garlics, those have historically been very rare, but we're now seeing more frequent outbreaks of those. So given these shifts, we wanted to explore if E. coli persists in soil amended with treated basalls, what are the chances of transfer? In our case, we focused on onions. So what are the chances of transfer to onions? And how do these environmental factors, such as rainfall or temperature, they affect its survival and potential crop contamination? So we'll go over those findings in the next couple of slides. So why did we do this research, right? We all know, again, Dr. Schneider has already set forth such a wonderful stage here. Fresh fruits and vegetables are essential for healthy diet, but as much they are essential, we are also having a hard challenge keeping them safe microbially. So in the past couple of decades, we've seen more and more produce-related outbreaks, and unfortunately, onions have also been part of that story. Now, we usually don't think of onion as a high-risk food, right? But historically, those outbreaks linked to these bulk vegetables were rare. But in the last few years, such as in 2020, there was a massive salmonella outbreak that was linked to red onions from California. A number of people got sick. More than 400 ended up in the hospital as well. And so the frustrating part is the investigators couldn't pinpoint a single root cause. Just some of the leading theories were maybe contaminated irrigation water. And they also found some other potential risk factors, such as always nearby livestock, lapses in packing facility sanitation. And just for an instance, last year in 2024, again, we had the 15787 that showed up in yellow onions that were served at McDonald's. So these outbreaks, they aren't just random one-off events. So we're seeing a pattern here. And that's why we wanted to dig a little deeper We already know that soil amendments like the composted poultry litter, heat-treated poultry pellets, they can improve the soil health. When used properly, the risk of pathogen contamination is also low. But what happens if something goes wrong, right? What if the amendments aren't stored correctly, or if there is an unexpected contamination event? How does the fate of enteric pathogens look like in such treated basalts? And if they do persist in the soil, could they transfer to the crops like onions? So these were the questions that drove our research, and we wanted to understand these key questions here. So this work was part of the CONTACT grant, which was led by Dr. Michelle Daniluk, who's at the University of Florida. And again, I want to take a moment to recognize the wonderful team that collaborated on this project, without whose hard work, this research wouldn't have come together. Special thanks to Bensi. Again, shout out to Bensi, who's now a PhD student at UGA, Cameron Bartsley, and of course, Dr. Schneider. We had a wonderful team without whose expertise we couldn't conduct this study successfully. So this study was conducted at a research farm, again, in Live Oak, Florida, where we set up the research plots. We had buffer zones around the plots in order to minimize any cross-contamination that could affect the validity of our results. And here is a visual of what the setup looked like. So as you can see, the plots were randomly assigned to different treatments. These colored flags, they indicate different treatments. And we also used sprinkler irrigation here, as you can see here, that irrigated each plot for about an hour a day. And we focused on 4 treatments in total. So number 1 was heat-treated poultry pellet, HTPP amended plots. The second was the composted poultry litter amended plots, and the rest of the 2 were positive and negative controls. So in addition to these soil amendment plots, we also transplanted onions into the plots, as shown in this picture to the right, to evaluate the potential transfer of E. coli from the amended soil to the crop itself. So for each treatment, we established 3 plots just as triplicates. And first off, here we have Dr. Schneider, who's applying the amendments here. So we applied the amendments at the rate of 680 grams per plot. And our goal here was to deliver approximately, to hit that nitrogen target to our soil, which is approximately 2% nitrogen to the soil. And as shown in this third image here, again, Bensi is spraying a liter of E. coli onto each of these plots to get a baseline of about 5 log CFU per gram of soil, mimicking a contamination event. So then finally, we have Cameron, who's tilling the soils from all 4 directions so that we uniformly mix the soil, the amendments, and we also made sure to sanitize those tiller when we switched between plots in order to prevent the cross-contamination as well. So once the soil were amended, inoculated, well tilled into the ground, we sampled the soils at specified intervals until over a 140-day period. And again, we were focusing on that in order to extend beyond the 120 days, which has been recommended by the USDA's National Organic Program. So our goal here, again, was to understand how E. coli survives over time in the soil, amended with these treated basalts in the sandy soils of Florida. So for our next research objective, which was to understand the transfer, after we set up the plots, we also transplanted onion seedlings onto these plots here. Once we transplanted those seedlings, we waited until after 133 days, where we dug up the onions from the soil. But before we just harvested them right away, we also let them go through the curing process for 14 days. So just to make it clear again, by curing, all that we're referring to is leaving the onions on top of the soil, exposed to the air, sun, just to naturally dry it out. After those 14 days, we harvested the onions and analyzed those samples both before and after curing to see not only the transfer, but also if curing also did any impact on the survival of E. coli on the onions. Now, with all these findings here, we are looking for if there is any practical implications for how we handle crops after they're harvested, whether curing might help reduce microbial contamination or not. And so we had some interesting results that I'm very excited to share now. So this figure here, it looks at the effect of BSAAO, basows, on the survival of E. coli during our field trials, and this is for the first year. So on the x-axis over here, we have the number of days we sampled, which runs up to 140. And on the y-axis, we show the level of E. coli we detected in the soil. So we use two different methods, MPN, the most probable number, and the log CFU method, in order to detect even the lowest level of E. coli in the plots. So just looking at the graph here, the blue line, it represents the poultry pellet amended plots. The gray line, it represents the composted poultry litter. The green one shows the positive control, which means there was no amendment at all, just a contaminated soil. And the yellow line, it represents the straight yellow line. It represents the baseline, the control, which has no amendments, no inoculation, just plain soil. So from the graph here, we can see that the positive control, that is with no amendments, which is the green line, right? Just a contaminated soil. It showed a rapid decline in E. coli within the first 14 days, after which it sort of leveled off or continued to slowly decrease. And this is what we would expect in an environment without any amendments that could support the survival of E. coli. It's like faster decline, and then it lingers on for a couple of days until it goes into extinction. However, in the amended plots, such as in this poultry pellet amended plots and the composted poultry litter plots, the level of E. coli, as you can see in the graph, it remained significantly higher than in the unamended plots. Interestingly, even among the two amendments, the heat-treated poultry pellet, the HTPP plot, had the highest survival of E. coli, with levels still at about, here you can see, about 1.5 log CFU per gram, even until 140 days, which is way above our limit of detection here, even with the spread plating technique. So this tells us something important here, that even though we expect that the treated poultry pellets, right, the heat-treated poultry pellets to be safe after the heat treatment, in case there is any contamination event, these amendments in the soil, they can prolong the survival of E. coli for a much longer period of time compared to the untreated soil. So while the pellet themselves might seem safe initially, they can extend the bacterial load in the soil if contamination happens. So how about year 2, right? So we repeated this experiment again in the 2022-23, running from November through April, to see how the results may vary over a second year. And our main goal here was to understand whether the environmental factors like temperature or rainfall would also impact the survival of E. coli in the soil, or if we would see the consistent patterns from year to year. So what we found over here was that the results were quite similar to year 1. As you can see from the graph, again, x-axis, y-axis mean pretty much the same. The different colored lines also represent the same treatments. So just it's evident that the blue line, which represents the heat-treated poultry pellet, it still showed that the E. coli levels were significantly higher compared to the unamended soil and even the composted poultry litter plots, even in year 2. So that was one of the repeated trend that we saw. But what is interesting is that after about 110 days, the bacterial levels, it started to decline in all of the plots, including the HTPP in year 2. So by the end of the study, even the E. coli levels in HTPP plot, as you can see here, it was much closer to the baseline level of the unamended soil or even the poultry pellet, poultry litter amended soil. So this particular trend was sort of a departure from year 1, where we saw that the E. coli levels were still higher until day 140, right? We still had about 1.5 log CFU per gram in year 1. So just over here as a conclusion, while the general trend remained the same, E. coli was higher in the E. coli was higher in the treated soil compared to the unamended soil. By the end of this second year, like we see here from 110 days and forward, Poultry pellet amendments, it did not keep E. coli at those elevated levels as they did in year 1. Poultry litter amended plot, again, it showed pretty much the same trend, and unamended soil also pretty much showed the same trend. So just to give you a clear picture again, between year 1 and year 2, we broke down the data for each of the treatments. And looking at the graph, you can see that the unamended plot, which is at this leftmost corner, which is just the contaminated soil, it showed a rapid decline of E. coli within the first about 14 to 28 days, but the bacteria still persisted at low levels after that. By about day 42, the levels were much lower than the amended plots. Now, if you look at the graph for the poultry litter amended and the heat-treated poultry pellet, which are the next two graphs, you'll notice that the E. coli levels in both treatments are consistently higher than the unamended soil. So this visually tells us something important, right? While amendments are beneficial for soil health, they can also provide a nutrient source for bacteria that we were talking about with the copiotrophic E. coli loving that nutrient-rich environment, which just allows them to survive at higher levels and for longer periods in the soil. So what's also interesting is that when we compare the poultry litter to the HTPP, poultry litter plots, it consistently showed a higher bacterial survival across both years. The poultry pellet plots, except for in year 1, where towards the end, year 2, it sort of tailed down, but in the year 1, the bacterial levels still survived. Now that was looking at the survival of E. coli just in the soil, right? Just in the amended soil. But what about the transfer from the amended soil onto the crop, specifically in our case, onions? And the key question we had was, did the bacteria make it onto the onions? And if so, did curing help in reducing those levels? So in order to achieve that, we sampled the onions both before and after curing within those 14 at 133 days and after 14 days, meaning at day 147. So we sampled onions to see if there was any change in E. coli levels on the crop. And these are some of our results here. So this table, let's take a closer look. It shows how much E. coli was transferred onto the onions before and after curing in both the years. So in both the years, we saw no transfer of E. coli from the unamended soil. 0 out of the 15 analyzed samples were positive, meaning there were no transfer of E. coli from the unamended soil, which is good news for growers, as it shows that unamended soil, it doesn't pose a significant risk of transferring E. coli to the crop. However, when we looked at the heat-treated poultry pellet plots, we see a noticeable transfer of E. coli from the soil to the onions, particularly in year 1. So in that year, we found that 11 out of the 15 samples that we tested, we analyzed, it tested positive. And in year 2, the number was much lower. We only had about 3 out of 15, but it still was there. So if you recall from our previous slide, we saw that in year 1, the levels of E. coli in that HTPP plot was also much higher, right? We had about 1.5 log CFU per gram, whereas in year 2, those levels, it significantly dropped. But as we see in this table, even though the bacterial levels were so low in year 2, they were still enough to transfer onto the onions. So what happened when we left for the onions to cure in the field for 14 days, right? So the results are pretty interesting again. The curing, it definitely helped reduce the risk. In year 1, the number of positive onion samples, it dropped from 11 to 6 after curing. And again, for year 2, it dropped from 3 to 1 positive onion sample after curing. So this tells us something important, is curing can be an effective practice for reducing that microbial risk in terms of onion production. It's a simple on-farm step, which does not need any post-harvest intervention, right? So it's something effective. So while we've seen a consistent trend where the E. coli levels were higher in amended soils in year 1, and those levels were lower in year 2, we started asking ourselves, what could be the reason for these differences? So to dig deeper, we started looking at the nutrient composition of the amendments, of the soil with those amendments, and how they affected the soil. And guess what? We found some interesting insights that could explain why the heat-treated poultry pellets behave differently in each of those years. So in this graph over here, it shows how nutrient levels in the soil were impacted by the addition of different amendments. On the x-axis here, we have different nutrient levels. On the y-axis, it's just the level of those nutrients. And as shown by these blue bars, for poultry pellet, we can see that when we added poultry pellets to the soil, the levels of key nutrients like the nitrate nitrogen, the ammonium nitrogen, the phosphorus, the potassium, they were significantly higher compared to both the unamended soil and just the poultry litter amended soil. So poultry pellets not only improved soil health because of the higher amount of nutrients, they also create that nutrient-rich environment, which we're talking about is that copiotrophic environment rich in nutrient, which helps in supporting these enteric pathogens to survive much longer and at much higher level. There are several studies that have also shown that E. coli can survive longer in these nutrient-rich environments, with nitrogen and phosphorus being some of the key nutrients here. And that's exactly what we saw in our study as well. So when we compared between the two amendments, HTPP are subjected to heat treatment. So just looking at how these are processed, HTPP is subjected to heat treatment, which kills most of the indigenous microflora in the pellets, which leaves only those nutrients remaining in there. On the other hand, we have composted poultry litter, which still contains a background microflora in there, meaning that there is beneficial bacteria that can serve antagonistic towards these harmful pathogens. So this could explain why in our study, the HTPP supported higher bacterial survival than the litter, because there is no competing microflora. Again, this is our hypothesis. There definitely needs to be much research done, specifically delineating these factors. But we hypothesize that the lack of competing microflora in those HTPP could help allow this E. coli to thrive, while the composted poultry litter, because of its natural microflora that's already there, it may inhibit the bacterial growth. In addition to the nutrient, we also looked at several different environmental parameters, and something that was striking was rainfall. So I'm just trying to show that graph over here for both the years. In addition to the nutrient content, rainfall may also have impacted is one of the other hypotheses we have here. So when we compare year one, which is the solid lines, as you can see here, and year two are these dotted lines, we can clearly see that year one experienced much higher rainfall towards the end of the experiment. And this is, again, a cumulative rainfall in every 10 days. And this increase in rainfall probably could be a key factor in explaining why we saw that higher level of E. coli, especially in year one. So the hypothesis is that the rain must have provided much needed moisture to the soil, which in turn can solubilize the nutrients that are already in the poultry pellet. Now, it does not have indigenous background microflora, tons of nutrients, and when there's rain, it solubilizes into the soil, and that makes the environment even more favorable for the E. coli growth. Again, this is a hypothesis based on what we observed, and we're still exploring how we can correlate that rainfall data with bacterial survival more rigorously, and more of these publications are coming in the future with some of the advanced modeling of the data with our collaborators from University of Georgia. But it's definitely worth noting that the higher rainfall in year one may have contributed to this longer bacterial survival, which we didn't see as strongly in the year two. So this, as part of this contact project, as Dr. Schneider mentioned earlier, we also replicated this study with Dr. Dunn's lab at Georgia, and we tried to see the same impact of different treatment amendments in the soil, and how location could also be one of the factors affecting it. I've also put a QR code over there if you're interested in diving deep into our paper. Please feel free to scan that. So just to give you a quick overview of some of the key findings from this study, we did the exact same methods in both of the locations. And what we observed was E. coli survived at higher levels in BSA amended soils, both in Florida and the Georgia locations. The soil properties were different in two of the locations. Georgia's soil was classified as loamy sand. We sent them for texture analysis, and the results for the Georgia soil was classified as a loamy sand. And Florida, the soil from live oak was classified as sandy soil. So despite having two different soil properties, it did not seem to influence the survival of bacteria. We saw the pretty much same consistent trends where heat-treated poultry pellets showed the highest survival, followed by the poultry litter, and then the least in the unamended soil. And we also found that the E. coli, it persisted up until 140 days, which is way beyond the 120 days of the National Organic Program recommendation in both of the amended soil at both the locations. And while we also looked at the weather factors, we did not find strong correlation between, and this was again only for the year 1 data, we did not take into consideration the year 2 data, and that is coming into publication. Bensi has been working on that, but just focusing on the year 1 data between those two locations, we did not seem to see any strong correlation between rainfall or temperature that enhanced the survival of E. coli. But from Georgia, we are doing some advanced modeling to get some key findings here. We just did some simple correlations, but more data analysis is ongoing on this work. So in our Florida field trials, synthetic fertilizers were also periodically added as part of the field management practices, which again isn't the common practice in organic farming. So we were curious how synthetic fertilizers might also interact because it was added in our field condition. We were interested in looking at how that also comes into play with the organic amendments. And so we did a microcosm study again. As Dr. Schneider said, lab studies are very different than the environmental studies, but we conducted this microcosm study where we took the soil from the live oak facility and recreated a controlled indoor environment condition mimicking the outdoor condition. The goal was to see what impact does the addition of this synthetic fertilizer would have alongside the organic amendments. And we saw some interesting results here. The soil with just the organic fertilizer, which is this green and then the red colored line, red being the poultry pellet, and then this light parrot green being the poultry litter. So soil with just the organic fertilizers, it followed the same trend earlier as we saw, HTPP showing the highest survival in microcosm, followed by the poultry litter, even more than the same treatment where synthetic fertilizer was added. So the bottom graphs over here, those are the amended soil with the synthetic fertilizer. But when there was no synthetic fertilizer, you can see the survival was higher. So this was a surprising finding to us. And honestly, we don't have a clear explanation as to like why that happened. But despite the antagonistic effect of the synthetic fertilizer, we still saw that the HTPP with the synthetic fertilizer here, it still was slightly, the trend was slightly higher. Even when we added synthetic fertilizer, HTPP was still showing that consistent trend of showing higher bacterial survival. But again, just as a disclaimer, the microcosm studies, again, these are controlled environments and they cannot fully replicate the actual field condition, but it's still a valuable insight as to what could happen if synthetic fertilizers were added to the soils along with the amendments with those organic inputs. So to wrap up our study findings, some of the key take-home points, BSAO amended soils, the amendments, it significantly influences the E. coli survival in soil, with the trend being highest in the heat-treated poultry pellet, followed by poultry litter, and then the unamended soil. We were able to detect these E. coli survival way up to 140 days in both the amended plots. Even when E. coli was present at that low levels in the soil, right? As we saw in the year 2, we only had, it went below the limit of detection of spread plate, and there was like very few cells left as detected by MPN in year 2. Even at that low levels in the soil, it still had the potential to transfer onto onions, as we saw 3 samples positive out of the 15 samples positive before curing, and then which dropped down to only 1 being positive after curing. So the good news is that curing it, which is like leaving onion above the ground for certain days, in our case, it was 14 days, it was effective in reducing the microbial load. And also when we compared between the 2 experimental years, year 1, it showed higher and longer survival of E. coli until the 140 days, and also a higher transfer of E. coli from the HTPP amended soils. And so from our hypothesis, which we sort of observed from the data that it could be attributed to the nutrient-rich environment, which is provided by the HTPP, and the high rainfall events that occur during year 1. But again, we have the modeling going on, and hopefully we can get that publication out soon, sometime this year. And with that, thank you all very much for listening.